Hello, everyone, and welcome to Opportunities and Challenges in Microbiome-based Clinical Research, presented by Medpace and Diversigen. I'm Molly Walker, Editorial Director at Fierce Markets, and I'll be moderating this webinar. Our speakers today are Brian Murphy, Vice President of Infectious Diseases and Vaccines at Medpace, Julia Cope, Director of Scientific Operations at Diversigen, and Stephen Johnson, Vice President, Regulatory Affairs, Medpace. You can read their full bios on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker to participate in the interactive parts of this presentation. If you'd like to download the slide deck, please click the Resources button at the bottom of your screen. This event is being recorded and will be available on demand within 24 hours after the event. We will follow the presentations with a Q&A session. Please submit your questions during or after the presentations using the Q&A box at the right of your window. Okay, now let's begin. Brian, please go ahead.
Thank you. And on behalf of Medpace and our partners at Diversigen, thanks everyone for joining this webinar on the opportunities and challenges in microbiome-based clinical research. My name is Brian Murphy. I'm the Vice President for Infectious Disease and Vaccines here at Medpace.
Stephen Johnson, I head up Global Regulatory Affairs and Medical Writing for Medpace.
Julia Cope, Director of Scientific Operations for Diversigen.
Very good. So we'll spend the next hour discussing a lot of opportunities that will come up in clinical trials, and I know many of you are joining this after you've seen some of our objectives. So just to remind you kind of an outline of what we'll be covering today. We'll be addressing some operational, scientific, safety, and endpoint challenges in trials evaluating the microbiome. We'll discuss how to incorporate microbiome analysis into your study, and this will be coming from our colleagues at Diversigen. We'll be discussing quality, specifically from a manufacturing to clinical trial aspect, and the CMC part of this will be fairly brief. And we'll discuss regulatory considerations at the end and some of the new regulatory requirements that are to be expected. So for the past 100 years or so, we've really been focused on a couple of dozen pathogenic organisms that have been known to trigger disease. And these were the ones that were quickly identified because they were the ones that could kill us and do so fairly quickly. But in fact, it wasn't until recently that only about 80% of the bacteria in our gut had ever been cultured. And it's only been even more recently that we've been to discover how the microbiome's networks of interactions affect us. So the microbiome, which is, of course, all the living cells that live in and on your body, plays a huge role in radically different areas of health, such as nutrition, early childhood, hygiene, infectious disease, and even chronic health conditions. Most of the time, we live in harmony or symbiosis with these microbes. There's only 3 pounds or so of it in the gut, but up to 6 pounds or so total in a 200-pound adult human. But they really do vastly outnumber us. The human microbiota consists of about 100 trillion microbial cells, again, primarily bacteria in the gut, and outnumbering us about 10 to 1, as you see here. In terms of genes, there are 20,000 or so human genes, but somewhere on the order of 2 to 20 million microbial genes. It's a wide range based on a lot of factors that we're going to be talking about through this presentation. So the idea that we are outnumbered and have so many more microbial cells than human cells, and that our health is a byproduct of these interactions with and between these organisms really runs counter to some of the long-held beliefs that our health is mostly orchestrated by instructions embedded in our own DNA. So now people begin to realize that other organisms, and some of these that have originally thought to have been innocent bystanders, have a more subtle way of interacting with us. Major conceptual breakthroughs in terms of our knowledge of what is normal and what is not has advanced, and our understanding of microbes as not only part of a human, but important in influencing our health and behaviors as human, and in turn are being influenced by our health and behavior, is becoming better understood. It's truly a very complicated and interactive ecosystem. So based on its size, organization, and specialized functions, many now consider the microbiome as a distinct organ, one that carries out activities essential to our well-being. So then what is normal flora? It sounds like an easy concept, but it's really sometimes quite difficult to understand. The dogma has been that we are born sterile, but this has been challenged with some of the advances in technology that demonstrates our early flora is strongly influenced by the way we are born. So normal flora does develop within the first few weeks of life and then takes up permanent residence. It becomes relatively constant after that, but that may be challenged by some of the factors you see on the slide here, such as the relative temperature or moisture in the environment where the organism exists, the age of the patient, the hygiene, even genetic factors such as what receptors, enzyme activity, or pH differences could be there, and multiple other unmeasured environmental exposures. So the dynamic nature of the microbiome partly explains the enthusiasm that surrounds it. There's evidence that dysbiosis in the gut is a factor in an array of conditions and diseases, some of which you see here on this slide. Recent advances in analyzing the gut microbiome and how it impacts immunity have put our ability to characterize and manipulate the microbiome within reach. Data from mouse models and even in our own clinics have demonstrated that the microbiome can even potentially impact immunotherapy effectiveness and may help us to understand drug toxicity. As we learn more about the human genome, scientists identify changes in the genome that increase the risk of disease, but it's really hard to overwrite some of those human genes or to find drugs that target them. But the microbiome, on the other hand, could theoretically be altered more easily through probiotics, fecal transplant, or other means. It is, as some researchers say, the only organ that can be replaced without surgery. So consequently, multiple clinical studies are underway that are testing single commensals, mixtures of defined species and subspecies, and cocktails of microbiota-derived molecules that are targeting specific microbial species or pathways that are either enriched or absent in a disease state in an effort to treat or prevent that disorder under study. So the role of the human microbiome as an agent of health and a potential avenue for therapeutic intervention is rapidly evolving and wide-ranging. For instance, stool transplants are well known to be a treatment now in Clostridium difficile. Numerous commensals are being linked with cancer. A higher abundance of Eubacterium lamosum has been associated with a decreased risk of relapse of disease after allogeneic hematopoietic cell transplantation. Early life probiotic supplementation has been associated with pancreatic islet cell autoimmunity. Engineered E. coli being used as a treatment for obesity. Targeted molecular inhibition of microbial synthesis as a treatment for atherosclerosis, and human vaginal commensal species like lactobacillus are being studied as a way to provide airway protection against allergen challenge and respiratory syncytial virus infection. There's a lot of active work going on in the microbiome outside the gut as well. Skin microbiota transplants are being used to treat atopic dermatitis, acne, and psoriasis, wound healing, and even cosmetic applications. Vaginal microbiota transplants to treat bacterial vaginosis, preterm birth, and pregnancy progression. Whole body microbiome transplants or partial restoration of the microbiota are being used for cesarean-born infants via vaginal microbial transfer. And then look at the relationship between the gut microbiome and the central nervous system. Human milk, oligosaccharides, and nutrition are being linked to axonal signaling and neuropsychiatric diseases and behaviors. There are studies looking at links between Bacteroides fragilis and anxiety. The lung microbiome in patients with cystic fibrosis is being explored more thoroughly, and oral microbiota transplants are being used for periodontitis or oral cavities. So clearly, the interest in the microbiome has surged over the past 5 years or so. The demonstration that fecal transplants are working in patients with Clostridium difficile infection and more recently in patients with inflammatory bowel disease lends credence to the field and to the hypothesis that restoration of a healthy microbiome can influence disease outcomes. These successes have ushered in a new area of opportunity for drug development in personalized medicine. On the slide, you see a look at the number of clinical trials and publications in fecal transplant and the microbiome between 2011 and 2016. Notice that the number of total trials reported on clinicaltrials.gov has increased from 0 in 2011 to close to 200 in 2016. The number that's continuing to increase and is probably likely underreported given that many trials running outside or exclusively outside of the United States may not be reported here. Likewise, the number of publications on PubMed where fecal transplant, microbiome, or microbiota are keywords has increased over 400% between 2011 and 2016. Clearly, there is growing interest in the microbiome, but still only a relatively small number of these trials and publications represent randomized clinically controlled trials. With the increase in interest in the microbiome, there has been an intense attention not only from researchers but from investors looking at business and partnerships ranging in size from small startups to global pharmaceutical companies. Multiple companies with therapeutics and clinical trials are really jockeying for position in a race to market, many with their lead candidate targeting Clostridium difficile and with a wide variety of other applications in the queue. Venture capital interest is mirroring this, with VC investment in microbiome companies growing at a faster rate than overall VC funding. Nearly $617 million has been raised for microbiome companies in 2016. It's a number that's more than all of the other venture investment in the microbiome space in 2011 through 2015 combined, an increase of nearly 460%. And it just started a few years ago, but the findings are truly so exciting it's triggered a cascade of investments. Now more than $1 billion total has been invested in the microbiome startups. So many of the areas are still in their infancy, but there are a growing number of clinical trials. Here's a map demonstrating the geographic distribution of microbiome clinical trials. As you can see, most of the clinical trials still involve the gut, with other category as in second place, and probiotic work in infant maternal health in third. So now that we know that microbiome trials are happening and where they are located, as you can see on this map, let's start talking about things about how analysis of the microbiome fits into them. Julia?
Here we show how the metagenomics can actually play a key role in investigations for therapeutics and diagnostic development. In early stage studies, 16S targeted amplicon sequencing is shown to actually have a difference between health and disease. The 16S is good to allow us to see taxonomy shifts only and is good for the genus level differentiation between health and disease. When we have found a signal between health and disease in the microbiome, we're able to take that further into WGS sequencing or next generation sequencing, where we look at the full genomic complement of every single one of the genomes in a sample. We're able then to look between health and disease at a functional level and at a taxonomic level. Then we're able to help design communities, consortia, single strains, or bioactives that are able to actually help attenuate the disease or impact the disease. Again, communities here are hundreds of strains that have been used in some therapeutics. Consortia are small numbers of very well-defined strains. Single strains, you get the point there, and bioactive chemicals that can be designed from those single strains or are produced by those single strains. The type of applications that are useful in this field include diagnostic applications of microbiome as an endpoint, probiotic or antibiotic effects on the microbiome, and we are now seeing patient stratification coming on, either patient stratification for clinical trials or personalized medicine in the field of immuno-oncology. Your microbiome, as Brian stated earlier, was thought to be completely sterile as you were unborn. However, recent literature has indicated that you are not completely sterile as you are born, and as you passage out into the world around us, you quickly gain a very full-fleshed microbiome. That changes throughout your lifetime, whether you are breastfed or formula-fed. You have a slightly different microbiome starting point. However, as we move into toddler, prepubescent, pubescent, and adult life, we have a very stable microbiome throughout our life that does shift over time, but is mainly stable. We're able to look at this through the spectrum of disease and watch an individual core microbiome, whether it be healthy or diseased, healthy or obese in this particular slide, have major blooms that we're able to find between health and disease. So the question early in the microbiome was, is there a single healthy, natural, normal microbiome? Here we're looking at 200 subjects in 16S space, or through targeted amplicon sequencing on the left-hand side, and through WGS sequencing on the right-hand side. With 16S sequencing, we're able to see that there is a large amount of variability in reportedly healthy individuals. And again, we're able to look down to the genus level. So that large blue swath are the Bacteroides. And if we're able to look a little closer through WGS, as in the graphic on the right-hand side, we're able to show that the single genera of Bacteroides has large variability at the species level and at the strain level. So normal, healthy, does vary quite a bit from person to person. Taxonomy alone cannot indicate healthy if you're looking to separate from disease. However, it can show you if there is a signal to find between those two populations. That is not to discount the fact that taxonomic levels don't tell us about the microbiome. We see here several different sites among the human body that have been tested for their taxonomic component, which is quite varied, and their pathway component, or the gene functions that are carried by those microbes. That particular core set of function in health is extremely stable from person to person and from body site to body site. So the devil is truly in the details in microbiome research. While we can find patterns in the taxonomic resolution, we often have to dig deeper into the actual functions that these microbes are doing or not doing, as it were. Here we're going to get a little deeper. As we see healthy or self-reported healthy, I want you to think about the idea that there is no single point in a continuum between health and disease where you flip from one to the other, but that there are many states of the human microbiome, some more healthy than others and some diseased and closer to disease than others. The challenge is to consider health and disease along this continuum and measure the entirety of the continuum and not single points. Here we're showing that healthy, as we examine it in mothers from trimester 1 through trimester 3 of pregnancy, they hold a core set of specific taxa in the beginning of pregnancy, and then at the end point in pregnancy, in that third trimester, we see the microbiome shift quite rapidly into a metabolic disease phenotype. So if you took patients that were undergoing regular metabolic disease or third trimester pregnancy, they would resemble one another better than the mothers had in the first trimester of this pregnancy. That is not to say that mothers in the third trimester of pregnancy are ill. However, they do have a microbiome that has shifted over to energy harvesting and scavenging so that they can supply the fetus. Would you not consider that to be a healthy stage for a third trimester woman in pregnancy? So now let's focus from the healthy microbiome into the diseased microbiome, and I'll hand it back to Brian.
We've already mentioned that many companies are using Clostridium difficile and in fact, recurrent Clostridium difficile as an entry point for a lot of different reasons. C. diff, as you are aware, may be present in the gut naturally in up to 5% of the population. It's transmitted person to person or person to fomite and then to person, so fomites such as bed linens or doorknobs. So it really makes it an infection control problem in hospitals or long-term acute care facilities. The pathogenesis occurs when normal flora is destroyed or altered through a number of mechanisms. C. diff then can overpopulate, and it leads to really a cascade of events that can culminate in intestinal perforation or eventual death. Treatment involves infection control procedures, removing the offending agent, and currently antibiotics are still the mainstay of treatment. However, these antibiotics are not always ideal as there are off-target effects, and the rate of recurrence is oftentimes unacceptably high. So consequently, fecal microbiota transplant has emerged as a potential therapeutic option through its ability to reestablish microbial diversity with beneficial gut flora. When we look at the states of health in terms of the microbiome, there are unique issues that arise in how the microbiome is manipulated and changes. Just as the states of health may not be binary, and the establishment of disease may be quite flexible, and the treatment of the disease may require a multi-step process, so too is it that the states of the microbiome are quite flexible as one moves on the spectrum from health to disease. So some questions remain. How ecologically stable is the microbiome during a patient's progression from health to disease? When does it stop being symbiotic and benign? Where are those tipping points when it becomes more characteristic of disease and less characteristic of health? How exactly is it influenced by the state of health that we are currently in? And how cooperative is it to change once we have started along this route to disease? There have been many parallels that have been drawn to the human microbiome from lessons learned from other ecosystems where balance is secured by the interactions, whether they be competitive or cooperative interactions among members of the community, and where this balance then determines the overall health of the ecosystem, or for our focus, the overall health of our patients. So similar to other ecological communities, a return to health and stability in a microbiome perspective is then time-sensitive and dynamic. An early and dramatic intervention that restores the pre-disease ecological interactions of the microbiome may be more effective in returning the disease state back to a healthy state. Late in the course of the disease, an intervention which could have been effective earlier may not be as effective. In other words, the damage that has already been done or resilience needed to heal is lost. Also, an intervention that's introduced later may have a higher hurdle to cross to restore the balance. The same intervention that may have been successful earlier may not have the sufficient interactions, either cooperative or competitive, to restore the natural ecosystem. Maybe perhaps because the organisms are now non-cooperative or are overly competitive. Whether the effect persists or not then is determined more by other non-intervention, non-controlled system factors, such as the immune system, other treatments that may be given to the patient, environment, or the disease state itself. Additionally, unpredictable and even negative effects in the context of a clinical trial, we would call those adverse events or serious adverse events, may occur through unsuspected networks and even may increase if the intervention is delivered later on the path. So in fact, the pathway from health to disease and hopefully back to health may be quite circuitous, with some of the most important outcomes, whether they be positive or negative, realized in the long term, emphasizing the importance of bringing in some long-term endpoints that need to be considered in chronic diseases and long-term safety issues that are not yet completely fully understood. Under the current disease paradigm, humans are healthy unless they have an illness that must be diagnosed and treated. As we've already discussed, a healthy, stable microbiome is one that balances both cooperative and competing networks of microbes, but it is not binary and exists in a very complex and dynamic state. Disease states will argue for the purposes of this presentation that the microbiome balance is off. If we use the example again of Clostridium difficile and the gut microbiome will become compositionally unstable and less diverse compared to what we would consider as normal, represented in the middle portion of the slide. The trigger for this disruption may be either an endogenous signal, so changes in host immune response that we might experience during aging or even diurnal rhythms, or there could be an exogenous factor, such as the mode of delivery for a neonate, the diet, the antibiotics that could be used, or other drugs, or even infection. When this disruption occurs, opportunists such as Clostridium difficile are permitted to colonize and proliferate, with the spectrum of disease, as we discussed, varying between asymptomatic carriage to severe complicated infection. To return to health or a state of stability or symbiosis, the balance between the microbes needs to be restored. In the context of Clostridium difficile, current research indicates that FMT is a product that can restore gut microbial diversity and can eliminate C. diff infection. For severe cases of recurrent diarrhea caused by C. diff, fecal transplants have been efficacious in about 90% of the affected patients that have been treated. Really, it's the prime proof of principle that healthy gut microbiota can reproducibly correct a severe and specific microbial dysbiosis. While recurrent C. diff is an obvious entry point, it's hardly the end indication for most companies looking to develop products that impact the microbiome. It has been shown that fecal transplant can also decrease the number of recurrences of C. diff. Recurrence of C. diff infection occurs in about 20% of patients after their initial infection, 40% of patients who have had at least one recurrence, and more than 60% after their second recurrence. How exactly fecal transplant cures CDI, or the mechanism of action, is not yet fully understood, but the data do show that fecal microbiota community is more dynamic within patients without recurrence and decreases in diversity with subsequent episodes of recurrence, as you can see in the graph here, moving from left to right. So acting through restoration of the ecological balance and diversity of the gut microbiota, particularly in terms of the restored balance, production of bile acids and short-chain fatty acids, as well as subsequent changes in innate and adaptive immune system, researchers believe FMT blocks the essential functions of C. diff, including sporulation, spore germination, vegetative growth, adhesion to epithelial cells, and even toxin production. So we've been talking a lot about fecal transplant, where fecal matter is the raw material for the treatment, but clearly there are other products that are under investigation that interface with the microbiome differently and bring in separate challenges and regulations. So a discussion about differentiating a drug from a dietary supplement will be important.
Thanks, Brian. So let's get back to some definitions for a moment, and I think this is essential as we look at developing or expanding our existing regulatory framework around these types of compounds. We can start with the definition of microbiome or microbiota. The microbiome are microorganisms, often a composite of microbes that inhabit a particular region or environment, which seems to be simple enough. Now let's add in a definition for something called probiotics, which we've barely mentioned here. Microorganisms, active microbes, or in some cases they're spores, introduced into the body for beneficial qualities. So the example that we have on the slide here is related to a specific probiotic used in many different examples, brewer's yeast or Saccharomyces boulardii. Now, most will agree that this microbe is a probiotic that has real or perceived benefits for humans. But what is it, or how might it be viewed from a regulatory perspective? Is brewer's yeast a drug, biologic, in a pharmaceutical sense, or is it something different? Is it a dietary supplement? And I think the simple answer is in how it's being described or advertised and its intended use. So if it's to be used by the general public without reference to prevention, treatment, or disease mitigation, then the case could certainly be made that the product is a dietary supplement, and that's what we would see in many cases. In this situation, a sponsor of a brewer's yeast product intended for the U.S. market would likely only need to be provided pre-market notification following the advertising rules of the FTC and the FDA. However, if one were to define this probiotic in the context of a prevention or a treatment, such as for recurrent C. difficile infection or CDI, then pre-market safety, efficacy, and approval would most certainly be required. With this in mind, then, we need to look at how to appropriately evaluate safety and efficacy for a given product or preparation in a specific and well-defined patient population. Meaning, we need to start discussing ways of approaching regulatory and clinically meaningful endpoints. And I think it's always important to take into consideration that just because something is clinically interesting or meaningful doesn't necessarily mean that it meets regulatory requirements for being regulatorily interesting. And so we have this discussion that goes back and forth sometimes that what may be appropriate for a patient and N equals 1 may not be able to be applied across a specific population. Okay. As we look at these regulatory and clinical endpoints, it's important to start to take a look at defining these more specifically in that we're looking at biomarkers, sort of either novel or exploratory or recognized for a particular disease state, taking a look at the possibility of using some of these biomarkers, if we understand them in more detail, as surrogate endpoints, and how they're linked with potential clinical outcome. Regardless, there needs to be a certain robustness to this endpoint, or any of these endpoints that we're looking at, if we're going to evaluate the probiotic in order to account for any of the total variability that we would see. And let's not forget that in most cases, it's not clear that a microbiome is actually causing a disease. And so it's looking at maybe some other aspects around this. Julia?
Here we're going to discuss briefly the microbiome destruction by antibiotic administration and the clinical endpoints that can be used, both typical clinical endpoints and possible microbiome endpoints. You can see on the top there are two different antibiotics that are administered. This is a preclinical assay in rodents. The microbiome is shown through the shifting graphs in both antibiotic 1 and antibiotic 2. Antibiotic 2 has a very large impact shown at day 1, where most of the taxa in the microbiome completely disappear and do not regain robust numbers until day 10 or day 14. In antibiotic A at the top, the microbiome impact is much smaller, and we regain robust numbers in most of the microbiome taxa by day 3 or day 6. On the right-hand side of the screen, we also show the clinical endpoint of C. diff colonization in the gut of those antibiotic-impacted animals. And we can see that one antibiotic has a much higher ability for C. diff colonization than the other. So this is a typical clinical endpoint for a current clinical trial. However, the restoration and the lapse in time in which the disruption happens and goes away can become a clinical endpoint, we hope, in the future. The metrics that surround this are currently the cutting edge of where we're working. Also to worry about in clinical endpoints is sustainability and engraftment of single-strain or multi-strain cocktails. To do this, you start with bacterial isolation, as you will need to produce the microbes for administration. We take those single bacterial isolates and we take different types of sequencing to get complete genome assemblies, whereby we can then begin to interrogate those genome assemblies, look at the gene content, the functional ability of those microbes, and very well characterize and describe those microbes. Furthermore, if you'd like to watch sustainability and engraftment of those microbes, we're capable of taking snippets of the DNA and making primers for qPCR or PCR assays from that bacterial genome, designing that so that you can have one single hit for your microbe of interest and verify that across all of the microbes of interest in a multi-strain cocktail. And then we're able to watch that move through the body, whether it's excreted or colonizes. We can watch sustainability and engraftment over time as it's excreted from the human body. This brings up the question of translocation and leads us into a place where we're talking about target populations and indications, where some of those target populations are more important for translocation than others. And I hand it back over to Brian.
Thanks, Julia. So we've already spoken quite a bit about the significant taxonomic inter-individual variation and the fact that the microbiota in each person is unique. So with all of that variation, is it possible to have a well-defined dysbiotic group? Well, the functional capacity of the adult gut microbiota with pathways involved in metabolism, fermentation, methanogenesis, oxidative phosphorylation, even LPS biosynthesis is relatively consistent across healthy people. So we believe it is possible to define a clear dysbiotic group, but the dysbiotic population needs to be one in which the microbiome has been directly linked to a disease, not just a weak correlation suggestive of the link, but causality is established or strongly inferred. Again, another reason that C. diff has become the likely entry point for many companies. While the primary endpoint should be related to established and accepted clinical disease endpoints and will likely follow similar prior regulatory approval pathways for similar indications, monitoring the effects on the microbiome may be useful for dose response, in other words, PK/PD parameters, or even as exploratory or secondary endpoints, which raises the question of development of biomarkers and surrogate endpoints. So this needs to be considered when identifying the most appropriate population. And in many indications, especially in chronic disease indications, the population needs to be also considered for its ability to provide long-term follow-up and comply with clinic visits to permit monitoring the microbiome over time. So microbiome analysis over longer periods of time raises the complexity and importance of appropriate sampling, the assays that are being utilized, and the timing and adequacy of samples for measurement, all details that rely on patients who adhere to a schedule of procedures. Not only is this of interest in demonstrating the sustainability of the microbiome's effect over short and long-term periods, but it is especially important from a regulator's perspective for long-term safety follow-up. Running global studies in these indications can be incredibly complex. We've discussed that the microbiome itself is a complex and dynamic organ. The products under study are quite variable and may be composed of thousands of microbes or just a single organism. Then there's the incredibly inter-subject variability with significant differences in ethnic backgrounds, diets, and numerous other environmental exposures. On top of that, the factors driving the endpoints are subject to major intra-individual variabilities that affect the microbiome that must be considered. Again, changes in lifestyle, such as diet, age, socioeconomic status, medications that could be used. All these factors can lead to data reproducibility issues and statistically underpowered studies where treatment groups run the risk of being significantly phenotypically, etiologically, and microbiologically different. With today's technologies, it is currently impossible to exactly measure fully the restoration of the microbiome for each individual, given that their entry into the study is often at a disease state. So it will likely be the first opportunity that their microbiome has ever been studied. As I mentioned earlier, clear, clinically meaningful human health endpoints that follow regulatory guidance will need to be used. To successfully achieve clinically meaningful endpoints, clinical trials need to overcome challenges in sample collection and extraction to make it practical for patients to participate. Patient variability, changes in the microbiome over time, differences in assays and measurements over time can make it really difficult to identify correlations and add to the complexity of establishing causality, or it can cause us to believe that we've detected a signal when there isn't really one there. All this variability confounding the endpoint makes understanding what correlates and what is causative to be not such a simple task, and is part of the reason that there are only a relatively few statistically powered longitudinal interventional studies evaluating the microbiome in patients with well-defined diseases. And without rigorous testing and randomized clinical trials, many will continue to question whether imbalances of gut microbial communities are a consequence or a cause of chronic disease. So how do we start to address the noise? Well, clearly a well-defined population is critical. As much heterogeneity as is in the system, clinical trials that are looking to achieve regulatory approval will need to closely weigh the risk-benefits for specific patient groups. Performing a risk assessment on each of how these groups may significantly add to the enrollment of the study, eventually benefit from the study, and contribute to the overall efficacy, needs to be weighed against their safety signal that they may generate. Obviously, in a large randomized clinical trial, the risk that each of these groups generate, in other words, measured in terms of the number of AEs, SAEs, or laboratory abnormalities, will, in theory, be balanced between treatment groups, such that there is no particular signal that is attributable to either arm solely from the background noise of that population. But with studies in earlier development, which are typically with smaller sample sizes, the risk for potential confounding or unequal distribution of a particular at-risk group to either arm will increase. For example, in a relatively small phase 2 study looking at FMT for patients with recurrent CDI, patients who are at risk for bacterial translocation through the gut, such as neutropenic or immunocompromised patients or even patients with severe GI mucosal damage, may be excluded. However, if you are developing a product to decrease the risk of relapse of disease after allogeneic hematopoietic cell transplantation, you're obviously including patients at risk for GVHD and patients who are immunocompromised. But you may consider excluding patients with inflammatory bowel disease or put restrictions on timing of prior radiation or even chemotherapy. Likewise, because of the potential for bacterial overgrowth in certain populations, such as diabetics with autonomic neuropathy and gastroparesis, a poorly controlled type 2 diabetic or one with autonomic neuropathy symptoms may be a reasonable patient to consider for exclusion in a recurrent CDI trial. But such a patient would need to be thoughtfully managed in making sure that there was balance between both arms in a study that was looking at the modulators of the microbiome and how they affect pancreatic islet cell insulin production in pre-diabetics or patients with type 2 diabetes. As safety is the other part of the risk-benefit assessment, studies need to be designed to ensure that short-term safety monitoring are properly collected and that the study is clearly designed to collect long-term safety data that continue to be a concern for the regulators. For instance, to address the potential for weight gain or loss after FMT, an issue which has been reported anecdotally and corroborated with some animal studies, clinical trials need to consider how weight and BMI will be captured and reported across multiple months. Deciding on whether to use a pre-illness historical weight or the weight from a calibrated scale on day one of a clinical trial as baseline is important. How do you follow this measurement over time? Do patients return to the clinic to use the same calibrated scale? Is the patient reported outcome reasonable? How do you analyze the data? Purely by measuring any change in the BMI or determining significance only when the BMI crosses from one category to another, for instance, from overweight to obese? Are body composition tests feasible and more accurate? These are just some of the details that will need to be considered. So with all the potential noise and confounders surrounding microbiome studies, patient stratification will likely be required to ensure efficacy signals are not masked and to help to balance the potential safety signals that may be seen. While stratification is important, there is a threshold where it no longer adds value. as in the case with many early phase studies that are typically smaller if there are too many strata in relation to the target sample size, and of course some of the strata could be empty or sparse. You take this to the extreme, and such that each stratum consists of only one patient each, it would in fact yield a similar result as a simple randomization with no stratification at all. On the slide here are some specific considerations for stratification for recurrent TBI studies, where successfully achieving the endpoint may be heavily influenced by and at risk from the number of prior recurrences, age, or even the infecting ribotype of the organism, and a host of other concomitant medications and procedures that are likely to accompany the management of this indication. Clearly, there are more strata here than we could incorporate into even a large study, but there are, however, comorbidity indices, which I'm showing on the right there, and scoring systems that we won't go into detail, but do combine some of these factors and may be a consideration for stratification and randomization, or at a minimum considered for collection of data for analyses. We're going to end with some regulatory discussion regarding issues that arise in studies evaluating the microbiome. And in the realm of biologics and fecal transplants, product safety is a particular concern. Many are still suspicious, and that many does include regulators, that transplantation carries the theoretical risk of transferring to recipients organisms that could become pathogenic based on certain host factors, or could transmit antimicrobial resistance to otherwise susceptible organisms. And there's reason for some of that suspicion. As many of you are probably aware, Jim was the name of a former milk wagon horse, and it was used to produce serum containing diphtheria antitoxin. After having been employed for some time, Jim eventually started showing signs that he had himself contracted tetanus, but unfortunately this wasn't realized until there was ultimately the death of a girl in St. Louis and 12 more children that were eventually traced back to Jim's serum, which was contaminated with tetanus in its early incubation phase. It's likely that these contaminated sera could have been easily discovered if they had tested this prior to use, but it wasn't done. There was no oversight at that time. Ultimately, this incident and a similar one involving contaminated smallpox vaccine led to the passage of the Biologics Control Act of 1902, which established CBER, and later in 1906, the formation of the U.S. FDA. Additionally, disease transmission by transplantation has long been recognized, including cases of rabies, Chagas, HIV, TB, and lymphocytic choriomeningitis virus. Furthermore, there are real, although they're not being quite realized, risks that transplanting fecal microbiota can spread infectious diseases such as HIV or hepatitis, as we saw in the 70s and 80s with thousands of people with hemophilia being infected with HIV from contaminated blood products. Even in the field of probiotics, there are cases of so-called healthy microbes turning rogue. We've already talked briefly about translocation, when those microbes in our gut cross the lining of the intestine into our bloodstream, causing a debilitating immune response and potentially sepsis. Linked to use of probiotics, there have been a handful of cases of lactobacillus species causing bacteremia in children with short gut or central venous catheters, and in 2 immunocompromised adults. Likewise, Bacillus subtilis bacteremia has been reported after a probiotic in at least 5 patients. While the most common adverse events are very transient and include abdominal discomfort and bloating, many regulators are concerned that there is still relatively little long-term safety data. For example, there are also theoretical hazards that FMT could change the microbiome to make people more susceptible to chronic conditions such as obesity or autoimmune disorders. FMT for CDI has been linked to relapses in inflammatory bowel disease and to the development of peripheral neuropathy, Sjogren's disease, idiopathic thrombocytopenic purpura, and rheumatoid arthritis. As we mentioned earlier, there's also been a reported case of the development of obesity following FMT from an overweight donor. I would say it's interesting that these same concerns could also be said for the use of antibiotics, which might also cause unknown lasting disruptions to the microbiome, but as of yet, antibiotics have not had to endure such scrutiny from the regulators. So how does one start to balance the risk? Part of the risk of FMT can be mitigated by mandating rigorous screening of the donor, CMC issues, which we'll briefly discuss next. A thorough characterization of the investigational product may be one of the most important steps to help to differentiate the product's role in a potential safety event of bacteremia, perhaps differentiating translocation or a suspected translocation from an investigational product from an unrelated infection to which the patient was at risk for non-FMT reasons. Additionally, sponsors need to ensure that the study sites are properly chosen and investigators understand the significance of organisms that may be cultured from various sites, especially in the urine and respiratory samples, where colonization needs to be differentiated from infection. While the evidence supporting the role of any particular organism in a pathogenic process seems obvious to perhaps this group here that's focused on the microbiome and infectious disease, it's not always as clear to all investigators and site coordinators in global studies, and becomes even less obvious in fields and indications beyond infectious diseases, such as diabetes, metabolic syndromes, or cancer. So clearly, rules are necessary and regulations need to be followed, but overly restrictive rules might encourage people to seek treatment outside the medical establishment. Instructions for do-it-yourself fecal transplants are available online. So clearly, the regulators understand the importance of ensuring reasonable pathways available for sponsors looking to develop products that address the microbiome, and in our experience, are eager to work with those developing products in this field. The concept of promoting balance, which is what many prebiotics, probiotics, and microbiome transplants claim to do, is not part of the disease paradigm that has governed regulation of health products in this country for years. But Stephen will talk about how that's beginning to change.
Fantastic. Thank you, Brian. So as Brian just mentioned in this last section, it's about 2013, FMT has really been recognized as a potentially viable treatment option for patients with recurrent C. difficile infection. And when I say recognized, I mean it's recognized by the FDA that it's and other health authorities that it's potentially viable. But it's important that the boundaries are always pushed and expanded in a safe and reasonably defined manner. From a regulatory perspective, the FDA recognized the need to put some guidance out there related to FMT that impacts both individual practitioners treating the N of 1 that I mentioned earlier and potential sponsors of therapeutic FMT preparations or products. So between 2013 and 16, the beginning of 2016, several formal and informal guidance pieces have become available. There's been different presentations given by the FDA and discussion fora that have come out. And in March of 2016, the FDA issued an enforcement policy regarding the need for INDs or investigational new drug applications for FMT. Specifically, this policy is to assure that patients with C. difficile infection not responding to standard therapies may have access to this treatment. So, as Brian mentioned, there's a lot of different ways of going that folks have looked at using FMT. Both sort of from a traditional therapeutic perspective and then sort of as an N of 1 as a practitioner, and the FDA stepped in to try to put some criteria around this. And so what they're looking for specifically is they evaluate whether or not an IND is required based on 3 primary criteria. First of all, the licensed healthcare provider treating the patient obtains an adequate consent from the patient or his or her legally authorized representative for the use of the FMT product. The consent should include, at a minimum, a statement that the use of FMT products to treat C. difficile is investigational, and there should be an adequate discussion of the potential risks and the benefit or the potential risk with this product. The FMT product is not obtained from a stool bank. This is where we separate sort of the N of 1 versus commercialization. And then item 3 regarding the stool donor and stool, that they're adequately qualified by screening and testing performed under the direction of the licensed healthcare provider for the purpose of providing the FMT product to treat his or her patient. Furthermore, during the period of this enforcement discretion that the agency is providing, FDA will continue to work with sponsors or individual practitioners as might arise who intend to submit INDs for the use of FMT to treat C. difficile infection, not responding to standard therapies. And as a final point for this slide, FDA intends for this to be an interim policy while the agency continues to develop a comprehensive approach for the study and use of FMT products under this IND. We've covered a lot of topics today, starting with very early discussions with regard to identifying areas of opportunity and challenges in microbiome-based research. It's a very exciting area. There's going to be a lot of successes in this area. We've already seen a lot of interesting programs move forward on the therapeutic space, and it will continue to move in that direction. It looks like there's a lot of investment in this area, which is fantastic. However, in order to increase the number of potentially successful clinical development programs in the microbiome space, this current area of state-of-the-art, it'll be important to recognize standard drug development principles and not overlook those with regard to global drug or biological development requirements. Oftentimes we look at the endpoint, look at all the sort of the fantastic ideas that are out there, and there are a number of good ideas and adequate or good approaches to address treatment of these patients. But it's important that we understand as we move from an N of 1 into broader populations, then standardization will ultimately be required. So understanding and having control over manufacturing processes and materials are really the first step in beginning to realize the potential for products and platforms. It's complicated. Biologics-based manufacturer requirements greatly exceed those for the manufacture of products like dietary supplements and other food types of products. It's important to realize that both from a manufacturing and a clinical perspective, growth media and host environment, respectively, may have a significant impact on the biological activity of a material in question or the product in question. Intrinsic and extrinsic host parameters and dosing regimens will have an impact on the safety and efficacy of a compound or preparation. And the question comes in, does one size fit all? This is multidimensional, so it's likely that it doesn't. Ideas that will need to be adequately addressed as we spend more time with these development programs. There's not a gold standard at this point. We need to continue to work at it. Finally, we come to long-term safety implications. It comes down to reaching agreement on risk-benefit. Brian, throughout the presentation, has talked about the concern of long-term health implications of changing one's microbiome, the impact of engrafting new cell types or new microbes into a donor. That's something that's going to be looked at, and will create a continually changing environment with regard to requirements. As we learn more and we're able to ask the right questions at the beginning of development programs and monitor the right things. We haven't really hit an APAC standard with regard to microbiome research to date, but with all the excitement from the patients, practitioners, sponsors, and regulatory agencies, success really seems to be within view, and the next 5 to 10 years will be extremely exciting.
Well, thanks, Stephen. Well, and thanks, everyone. It concludes the presentation, and I know many of you have been entering questions as we've been going through, so we've been collecting those. If you do have questions, please continue to submit them. We have about 8 more minutes right now in the hour for the presentation. We will get as many of these questions as we can, and those that we're not able to address just due to time constraints, we'll try to follow up with you later on. So please go ahead and continue to ask your questions. So I'm going to start, and I'm going to take the liberty of trying to group some of these together as they've been coming in. A lot of these are under similar categories, so I'll be paraphrasing some of your questions, but I think we'll hit many of these. So the first, I think this is a question for Julia and Diversigen. Can you differentiate the value added in performing 16S versus whole genome shotgun sequencing? Mouthful.
Definitely. So the difference between 16S and whole genome shotgun sequencing is that 16S is very targeted, it's quite quick, and it's inexpensive to run. It gives you the ability to look between health and disease or between dosage schedules and see if there is a major change in the community of the microbiomes of those patients. Whole genome shotgun sequencing allows us also to look at the functional complement within the species and strains that we're able to identify. We get more genomic information from a wider range of the community. 16S, the value is that you can see the entirety of the community with the depth of the sequencing from an amplicon. And WGS provides support for function and deep taxonomy identification. So they both add value, and right now, both are actually recommended for early investigations. After you've discriminated a single target or a group of targets in the type of study you're doing, we can really help you hone in on where you want to spend your budget, really getting a good answer to what is the functional characteristic of that community that's actually causing the effect you're seeing.
Great. Thanks, Julia. We have a regulatory question, I think, for Stephen. What would need to be done from a regulatory perspective to satisfy the requirement for changes in the microbiome to actually be a primary and regulatory acceptable endpoint instead of a secondary or exploratory endpoint?
That's a great question. I think we're probably. Thank you, Brian. I think we're a ways away from that, getting to that, where it's used as a primary endpoint. I think right now we're looking at usually the primary endpoints right now for these trials are looking at clinical outcome and not looking at these as surrogates. That being said, if we're able to identify, or sort of being able to link causality with outcome and through a surrogate, a surrogate or a marker, if you will, then I guess there could be a potential for that. Julian?
I definitely agree with you that secondary endpoints are currently where we are, not primary endpoints. But to say it's a surrogate for disease, the best place we have right now is the dysbiotic or non-dysbiotic microbiome. So that would be a diversity measure change, and you'd actually have to look at a change over time to know early on in treatment and later in treatment or pre-treatment and post-treatment whether or not you were having a change or an effect in the microbiome. As we've said throughout this set of slides and this webinar in general, the healthy microbiome, though it can be contrasted against the diseased microbiome, does not have a single state at which it is healthy.
As we were talking earlier, surrogate endpoints really seem to lend themselves to certain fields more than others. Oncology, for instance, perhaps more than infectious disease. There are some examples of surrogate endpoints in infectious disease, HIV and viral loads, hepatitis C, viral loads, or fibrotic markers perhaps there. But especially in the world of, so I think it may be a discussion that is very indication specific and may be variable depending on if it's more oncology or something that has more traditional or more acceptable pathway where surrogate markers have been used.
And it's part of its understanding whether sort of the impact of the does the microbiome or the change in microbiome cause the disease, or is there some other feature there? And so that's, at the end of the day, that's kind of what we're looking at.
Yes. What's cause and what's effect, right?
Exactly.
I think we have a question or series of questions for Julia again. What approaches are available for interrogating single strains for the purpose of characterizing organisms that may be translocated?
Okay, so interrogating single strains for translocation, the first thing, you need to have a single strain identified, and you have to have it characterized quite well to be able to follow it during translocation or the movement from the original site to secondary sites, whether it be blood, through the gut, or into wounds, if it's a skin or a topical strain. To interrogate that strain, you're really going to need an entire genome of your strain. Again, you're going to want to characterize it well before you turn it into a drug product. So taking the entirety of the genome, we are capable now of doing long sequencing reads that will allow you to have a full genome that has been circularized. You can then annotate and know the functions that the genome is fully capable of, whether or not there are plasmids or any other worrisome sections such as prophage integration or antibiotic-resistant genes. That gives you the full information on your single strain, and translocation can be completed through PCR or qPCR in the main tissue or introduction, whether that be oral or fecal, and also in the case of gut, leaky gut, you can watch that either appear in the blood or hopefully not, as it were, and watch over time whether or not you have colonization of that gut, and if you continue to see the microbes turning over and colonizing within. So the best approach right now is very good detailed description and following through qPCR or molecular means.
Okay, thanks, Sheila. I think we probably have time for one more question, and this one is a regulatory question that was specific about looking at how the FDA has determined that it's going to exercise enforcement discretion regarding IND requirements for FMT to treat C. diff infection, and particularly looking to see if there was any discussion around the rationale that the FMT product, that the FDA would exercise enforcement discretion when the FMT product is not obtained from a stool bank. So, Stephen, any insight into that?
So when the product's not obtained from the stool bank,
yes.
It comes down to it comes down to the understanding of the stool donor and the material that's going to be used and how that's characterized. The challenges with using it from the stool bank at this point in time is still around safety, overall safety. But when we talk about doing it for an individual, and we want to make sure that if somebody's at the end of the treatment, sort of their treatment paradigm, and there's nothing available for them, that there was an availability of that material for treatment for those patients.
Okay. Well, great. I will just say there are a lot of other questions that have come in, and I think we're out of time and we won't be able to answer those. We will try to, as many as we can, follow up offline with you if we have your contact information and provide you some answers to that if it's acceptable. So thank you for your time, and we appreciate your listening to this presentation.
Great. Thanks, Brian, and thank you everyone for attending this Fierce Markets webinar and submitting so many great questions. I'd like to thank our speakers for participating and Medpace and Diversigen for presenting today's webinar. This webinar has been recorded, and you'll be able to access the recording within 24 hours on the same page you used to register for this event. Thank you again for joining, and we look forward to seeing you at future events.